Thank you all for being here. Today, I will be talking about integrating single-cell and plasma proteomics in order to predict surgical site complications. My name is Amy. I'm a fourth-year medicine at UC Davis, and I'll be presenting my research with the Godelier Lab at Stanford School of Medicine. I have no financial disclosures. Complications occur approximately 1 in 5 surgeries. That means of the 110 million surgeries performed each year in the US and in Europe, there are approximately 22 million complications. Of that, approximately 2.2 million are surgical site complications, and that translates to prolonged personal suffering and significant socioeconomic burdens in the form of longer hospitalizations and readmissions. Postoperatively, a patient's recovery trajectory is complex. That means despite very similar demographics and patients receiving similar surgeries, the development of these complications can vary quite drastically. The current surgical risk calculator, NISCIP, predicts surgical site infections with an AUC of 0.7. For other complications, such as reoperation or readmission, it's not adequate, so an AUC of less than 0.7. As such, perhaps we can integrate the biological parameters that drive the pathogenesis of surgical site complications in order to increase risk prediction accuracy and improve patient care in the long run. Within minutes of surgical injury, there is activation of the inflammasome that leads to a TLR activation by DAMPs, and this results in the secretion of pro-inflammatory cytokines such as IL-1 beta, IL-6, and TNF-alpha. IL-6 in particular activates the STAT3 pathway, and it has been shown to forecast adverse postoperative outcomes, including onset of delirium and mortality. Simultaneously, there is an anti-inflammatory and immunosuppressive adaptation. So dysregulation in either direction increases the risk of postoperative complications, as overt inflammation can lead to SIRS, while profound immunosuppression can predispose a patient to developing post-surgical wound infections or sepsis. The immune response to trauma is very well orchestrated, and it virtually involves all branches of the immune system and engages all these immune cells. Therefore, it's important to be able to study the immune system at a single cell level and understand the function and role these cells play in the development of adverse outcomes. Luckily, this type of high-resolution study is now possible with mass cytometry. So using up to 50 phenotypic and functional markers, we're able to classify most of the major innate and adaptive immune cell subsets and quantify their abundance. We can then look at each of these populations in detail and study their function, so their endogenous response or how they can respond to cytokines such as LPS, interleukins, or interferon alpha. That we can then form a network of all of these immune cells to understand holistically how the immune system responds to immunological insults such as surgical trauma. This brings us to the goals of taking this technology in order to improve predictive performance. So we hypothesize that single-cell and plasma proteomic elements of a host's immune response to surgery can accurately identify patients who will develop a surgical site complication after major abdominal surgery. We recruited 41 patients for this study, and 11 patients developed a surgical site complication, which is defined as either a surgical site infection, anastomotic leak, or wound dehiscence within 30 days of surgery. Blood samples were collected before surgery as a baseline sample, as well as on postoperative day 1. The whole blood samples were either left unstimulated for endogenous activity or stimulated with cytokines, and then analyzed using mass cytometry. In parallel, 272 proteins from the plasma were quantified using Olink. So mass cytometry produces a high-dimensional data set with very complex and interrelated processes that require advanced machine learning methods. In simple terms, we created elastic net models for each of the 9 data layers in order to predict the development of a surgical site infection. We then integrated them using a stack generalization method in order to yield a final predictive model. This model classified patients with a high accuracy, so an AUC of 0.86, and it remains predictive of surgical site complications when accounting for patient group differences such as surgical duration or use of biologics like TNF-alpha inhibitors. To delve into the biology a little bit more, certain pro-inflammatory proteins were elevated in patients who develop a surgical site complication. This includes STREM1, which is an enhancer of the TLR4 pathway, as well as LIF, which is an IL-6 family cytokine. Other proteins were actually decreased in patients with a surgical site complication. And this includes EIF5A, which is implicated in T cell activation and autophagy, as well as KRT19, which is involved in epithelial cell differentiation, particularly of the colonic mucosa. And this suggests that patients at risk of surgical site complications may have impaired healing. I'd also like to highlight a couple specific immune cell features. So consistent with prior findings, STAT3 activity in classical monocytes is activated in patients who will develop a surgical site complication. Remember that the STAT3 pathway is activated by IL-6. So when coupled with the elevated LIF levels in these patients, this suggests that activation of the canonical pro-inflammatory pathways is more pronounced in patients who will develop a surgical site complication. Additionally, the abundance in the STAT3 signal was elevated in immunosuppressive MDSCs, which suggests that unchecked systemic activation of an anti-inflammatory immune cell subset in response to surgical trauma can contribute to the development of a surgical site complication. And this is reflected in the adaptive immune branch. So in summary, the complex interplay between pro-inflammatory and immunosuppressive cellular mechanisms after surgery differed between patients who develop a surgical site complication from those who don't. This difference is detectable as early as 1 day after surgery and is harmonious with current theories on surgical site complication risk factors. However, we had these baseline pre-surgical samples and we wanted to ask the question, can we predict if a patient will have a surgical site complication before surgery? And the answer is yes. So we applied the same analysis algorithm to the baseline samples collected before surgery, and the resulting model accurately differentiated the two patient groups with an AUC of 0.82. This model actually improves to an AUC of 0.94 when integrating parameters from the ACS risk calculator. This leads us to our current clinical trial on prehabilitation, where patients were recruited and enrolled in a rigorous 3 to 6 week prehabilitation program in which their fitness, nutrition, meditation, and cognition were optimized. Preliminary data from this study has shown improvement in some surgical outcomes. So in conclusion, CyTOF analysis revealed key immune responses that mediate important adaptations in the development of surgical site complications. Multivariate analysis was able to produce models more predictive of these complications than the current gold standard. The models remain significant when accounting for confounding factors. And finally, this is a promising avenue for guiding preoperative interventions such as prehab or surgical timing. Finally, I would like to thank the amazing team that I had the opportunity to work with in order to make this study happen. And thank you all so much for listening today.